All right, so these are the three mechanisms that are responsible for keeping GFR at 125 if our mean arterial pressure is anywhere between 80 and 180. Now, outside of that, we're talking about differing physiologic situations such as injury or hemorrhage or dehydration. And so the kidneys cannot hold the GFR at 125 beyond those circumstances, okay? But as long as we're between 80 and 180, these three mechanisms are what keep our GFR stable. So the myogenic regulation is the first mechanism, and this has to do with the smooth muscles in the wall of the afferent arterioles. So very similar to what we saw with our cardiovascular system, myo meaning muscle. And so the muscle, the smooth muscle that's lining the afferent arterioles specifically, so only the afferent branch. And here again, I'm going to point out that the afferent arterioles have a larger diameter than our efferent arterioles. So the smooth muscle that's lining this afferent arteriole is sensitive to pressure. So think about baroreceptor responses. So when there's excess blood pressure and we have a stretching of these smooth muscle cells of this afferent arteriole, the response that's brought about is to contract. Okay, and this should sound very familiar to what happens in our vessels, in in the cardiovascular system that we talked about. So when we stretch these afferent arteriole smooth muscle cells, the automatic response is for them to constrict on themselves. And that is a protective mechanism to keep GFR stable. So although I'm getting more flow, I'm gonna keep GFR the same by constricting on these, the afferent arterioles. The second regulatory mechanism, or the second intrinsic regulatory factor, is tubuloglomerular feedback. So this happens because of a group of cells that are located here in the ascending limb of the loop of Henle. Now, some texts will tell you that it's the ascending limb of the loop of Henle. Some texts will tell you that it's the distal convoluted tubule, so the DCT. There's a lot of debate as to exactly where these cells are, but we know that they're present along the tubular structures. So the tubular structures are going to sort of fold themselves to come in contact with the glomerulus, okay? This is going to be a little bit weird to kind of visualize. Let's go back to this slide quickly. Here. So as we can see, the glomerulus is here. It comes into contact with the distal convoluted tubule. So this is the proximal convoluted tubule. This is our loop of Henle. This is our distal convoluted tubule. And can we see how the distal convoluted tubule kind of comes adjacent to the glomerulus in between both of these capillaries, the afferent and the efferent arteriole? And that is so that the cells that are present in these this part of the tubular structure can sense the change that's happening in the rate of flow and can communicate back to these afferent arterioles. Okay, so just kind of bear this relationship in your mind as we speak about the tubuloglomerular feedback. So here again, we can see that much more closely. So we've got the afferent arteriole, the efferent arteriole, and here is that part of the distal the distal convoluted tubule or the ascending limb. Again, some books will tell you either of those, but basically there are cells that are present here. These are called macula densa cells, and they are determining how much flow is coming through this part of the tubular structure. And so if you've got a lot of flow that's coming through, these cells will send paracrine signals, to the afferent arterioles and tell them to constrict or contract. And so the smooth muscle of the afferent arterioles are basically responsive to those chemical messages that are coming from the macula densa cells. So if the macula densa gets a lot of flow, it's going to tell the afferent arteriole via chemical messengers, which we call paracrine signals, to constrict those smooth muscle cells, basically decrease the amount of blood that's coming across the filtration membrane, to likewise decrease the amount of flow that's coming through the tubular structures. okay? And then our third mechanism of action is brought about by the mesangial contractile cells. And I like to compare the mesangial cells to the precapillary sphincters. So if you remember the precapillary sphincters that we talked about back in the CVS system, they were basically these strips of smooth muscle that were around the capillaries, and they were helping to regulate the flow as well. So whenever there was increased flow, if there's increase in flow coming through the glomerulus, these mesangial cells, which are basically smooth muscle cells, are going to constrict to decrease the flow that's coming through. And the opposite is true. If we have decreased flow coming through the glomerulus, coming across the filtration membrane, these mesangial cells will relax in order to bring more flow in. So if you understood the precapillary sphincters and how they act, you can compare that action, that response, to the mesangial contractile cells, okay? And so these three mechanisms are regulating GFR, regulating the amount of flow that's coming in through the afferent arterioles. And I want to make the connection here. If we've got more blood coming into the afferent arterioles, we have a higher GFR. We have a higher rate of filtration. If we have less blood coming into the afferent arterioles, we have a less or a decreased rate of filtration. And so the three mechanisms that I talked about are really responding on to the radius of the afferent arteriole. So if there are signals that are telling us the GFR is increasing, all we do is decrease the radius by contracting the smooth muscles of the afferent arterioles. That'll decrease the amount of blood coming in, which will decrease our GFR back to where it needs to be, 125. And if the opposite is true, we see the opposite as well. So if we have more blood coming in, if we have less blood coming in, we're going to send signals to increase the radius so that we can get more blood coming in, so that we can rise our GFR to keep it at 125, okay? All right, what questions do we have at this point? Are there any questions here? Okay, I don't see any questions coming in. So I'm gonna just go through, and this flow chart kind of summarizes those two of those mechanisms. So the myogenic regulation and the tubular glomerular feedback. So I kind of walk through this, and hopefully if there were any confusion or confusing areas, then that'll be clarified here. All right, so our initial response is that, our initial response is that we have an increase in our mean arterial pressure. Think about an increase in blood pressure. So an increase in the flow that's coming into our afferent arteria. So that's our initial response. Now, we don't want GFR to increase just because we have increased flow to the kidneys. And that's what I mean by heavily regulating that GFR. We want GFR to stay at 125 milliliters per minute. So in order to do so, I'm gonna have to bring about some mechanisms in order to keep GFR stable, even though I've got more blood coming into the kidneys. So when I increase blood pressure, that is going to increase the stretch on my afferent arterioles, or the smooth muscle that's lining this arteriole. That is going to bring about a response of constriction of that smooth muscle, which will increase the resistance or decrease the radius, like the cross section or the diameter. That is going to in turn decrease the capillary or the glomerular capillary pressure. So remember, if I constrict the smooth muscle of my afferent arteriole, I'm essentially decreasing how much pressure, how much flow is coming into the glomerulus. Now, when I do that, that's going to give a negative feedback response onto this increase in glomerular capillary pressure. And so that will keep my glomerular filtration pressure up, and that will keep my GFR up. So it'll stay at a steady, constant rate instead of increasing excessively, which is what we do not want in the face of increase in mean arterial pressure. So this first mechanism is a way to keep my GFR stable even though my mean arterial pressure increases. And the opposite is true. So remember, this is happening in both directions. If I drop mean arterial pressure, I am going to bring about the opposite set of sequence of events, right? I'm going to decrease my resistance. I'm going to relax that smooth muscle layer, and that is going to seek to increase the flow, which will keep my GFR stable, okay? And that's the first mechanism. The second mechanism we talked about was that tubulo-glomerular feedback. So if we have an increase in our pressure, we increase the amount of glomerular capillary pressure, so essentially the amount of blood that's coming into the afferent arterioles. That will increase our glomerular filtration pressure, so how much blood is being filtered across the glomerulus and into those tubular structures. And that will increase my GFR. Now, in order to regulate this, the macular denser cells are going to be detecting an increased flow. Remember, if I'm increasing how much filtration is happening, I'm going to increase the amount of flow that's going through my tubular structures. And the macular denser cells will pick up that increase in flow, and they will release that paracrine signal, which are basically chemical messages that will tell the afferent arterioles to constrict, right, to contract that smooth muscle, which will decrease the resistance, and that will decrease my glomerular capillary pressure or decrease that amount of blood that's coming into the afferent arteriole. And that'll be a negative feedback to turn off this initial signal. Okay, so both of these mechanisms work in conjunction to keep our GFR pretty stable in the face of changing mean arterial pressure, but it's only within that window of 80 to 180, okay? All right, any questions on any of that? Okay. Now, I want you to take some time as homework before we meet again, and I want you to describe the 3 processes of intrinsic regulation of GFR. So when we start next time, we'll start with that, but try to pen down an answer that expresses a response to this question. So describe the 3 mechanisms of intrinsic regulation of GFR.